Right, so picking up from where we had our class cut off the last time, we are talking about the ester bond in lipids. Just as a
Yeah.
quick recap, lipids are organic molecules. Just get the highlighter there. They're organic molecules, they are vital for life. They form structures such as the cell membrane, hormones, they are energy stores, and they also have physical roles, such as our lips are primarily fats. They are obviously they are made up of carbon and hydrogen because they are organic molecules. There is very little oxygen. There's basically oxygen in the carboxyl group of the fatty acids and the OH groups of the glycerol, right, and fatty acids react to form lipids. There are two types, oils and fats. We think that they're one and the same, but it's basically the source and how they are at room temperature. So oils are sourced from plants and they are liquids at room temperature, whereas fats are sourced from animals and they are solids at room temperature. The lipids in biological systems, right, and in human beings are between 15 to 17 carbon atoms long. They could be different lengths, but this is like often, so generally between 15 and 17 carbon atoms. Uh, right, they may be saturated or unsaturated. So what's the difference? If all the carbons have single bonds and they're forming 4 bonds each, then it's a saturated right fatty acid or lipid. If there is at least one or just one, in fact, double bond in the chain, that is a monounsaturated. So having double bonds makes it unsaturated, and you can understand that the saturation is being talked, that's being talked about over here is the number of carbon bonds, or number of bonds that carbon is forming. So if it's not forming bonds to its full potential, that makes it unsaturated. If there's only one, it's called monounsaturated. If there's more than one, it's called polyunsaturated. So you can write something like that. And it also affects its physical properties. So saturated fats tend to be solids more, whereas unsaturated fats tend to be more in the oil category. Then we came to so they're made up of fatty acids and glycerol. This is a molecule of glycerol, so it's 3 carbons, right? And each carbon has an OH group with it. So it can potentially bind with 3 fatty acid molecules. And this is the general structure of a fatty acid. So you've got the acid part, the carboxylic acid part over here. The R group represents the carbon chain, which could be any length. And so the OH from the fatty acid and the hydrogen from the glycerol, they react. Water is given off, right? It is removed. So it is a condensation reaction. Let's get the pen. And then what you end up with what's of interest is over here. So this is the ester bond, right? And so you can potentially, in fact you do, what we have in our body are triglycerides. Right, and so triglycerides basically meaning that you've got a fatty acid chain attached to each carbon of glycerol. Right, and so that means a triglyceride. So right, another fatty acid chain over here, another here, and so you've got 3 of those. That makes it triglyceride. And so you've got those in the body. It's not necessary to be that way, but right, often is. And obviously this is a reversible reaction, so right, you take it the other way, and that is hydrolysis. So obviously, right, lipids are digested in the body, and so they're broken down into their constituents, fatty acids and glycerol, right? The addition of water, and that would give you its components back. Achha, anything that we're missing? Let me check. Right, so as I mentioned, saturated fatty acids are more likely to be solids at room temperature. Unsaturated fatty acids are more likely to be liquid. And the longer the chain, the more likely it is to be a solid. Okay, so obviously, the longer the chain, the more the mass, the more the molecules, right? The more likely it is to be a solid. And that wraps up that little bit from the lipids. Any questions?
No, sir.
Okay. So now we move on to proteins. The fun guys. Like fun guy, not like the organism.
Yeah.
Okay, this is chapter 1A5. Right, and proteins are, they're macromolecules, so they're huge molecules, and that means they are polymers. They are made of the amino acid monomer units. They contain, again, primarily carbon, hydrogen, oxygen, right? Because they're amino, they have nitrogen, and right, there are certain groups attached to each amino acid, in which case, right, actually there's just like one particular one that has sulfur along with it. Okay, so they're called amino acids, and so what they look like, you have, let's say, right, we've got a carbon molecule here, and the way it's written is NH2 and a COOH, a hydrogen here. So we can recognize, right, this is an amine group, right, this is a carboxyl group. So amino acid, so amino and acid, so that's what's giving it its name. It has both the amine groups and the carboxyl groups. Hydrogen out here, that's always present. So these three, right, are always present in every amino acid, and what really makes the difference is the R group, right? So the R group can be something simple, like a simple hydrogen, and could be something like, cyclic structures, or even double, you got two cyclic structures attached. And so the difference in the R groups is what makes different amino acids, and there are 20 naturally occurring amino acids. Right, again, depending on the R group. So 20 different R groups, 20 naturally occurring, right? Naturally occurring meaning they are sourced from nature, and right, as we know that it's basically plants that are the source of nutrition for the ecosystem. So they trap energy, photosynthesize, they get nutrients from the soil, water, right? And so they make the carbohydrates and the fats and the amino acids, and then the polymer structures. Uh, and so right, plants are able to manufacture all 20 of them naturally, right? Animals, or what was the term we used, autotrophs and heterotrophs, right? So the plants being autotrophs, right, can manufacture all 20, and the heterotrophs, right, they obtain it from other sources. Right, so for us humans, having a balanced diet of plant and animal origin, meat and milk and eggs and all sorts of fruits and vegetables and bread, and that would ensure that we have a good supply of these amino acids in the body to make proteins, because proteins are everywhere, right? And DNA basically codes for proteins, but not going too far ahead from the contents of the chapter, right? And so just to they've given some statistics in the book, right, that 18% of the body is made up of proteins. All your enzymes are protein, your hair, skin, nails, right, all of that is protein. Hemoglobin is the protein that carries oxygen in the blood. So you can understand, right, how vital that is. We've done the basic structure, and then let's talk about, right, the formation of proteins from amino acids. So keep in mind that proteins are the macromolecules, and the monomers are amino acids. So amino acids are what make proteins, but they are not synonymous. You cannot interchangeably use amino acid with protein, right? So once I discussed the bonding and the structure, there's another term you can use for proteins. I'll mention that, not just yet. So, right, in carbohydrates you had monomers of monosaccharides linking up together to form polysaccharides. Lipids were different because you had two different molecules joining up, glycerol and fatty acids, to form the lipids. In proteins, again, it is technically the same monomer units. The R group is different, but you've got amino acids joining up to form protein. So the structure, let's take two amino acids generic. We're not going to go into details of the R group, and let's draw them out. So you've got the carbon, you've got the amine group over here, and a hydrogen. R group number 1, then the carboxyl group, and then you've got another one over here. Right, and so you've got two of these amino acids, R2, just to distinguish. And of course they could be exactly the same, they could be different, but that is not the point at the moment. And the way they're reacting, right, so again you've got two of these molecules coming together, it will be a condensation reaction, and these are the atoms involved. So you're losing water. It's a condensation reaction. And what you're ending up with is the peptide bond. And so this over here is a peptide bond. Now again, obviously it's a reversible reaction. The reverse would be called hydrolysis, and we'll be adding water in that case. Right? When saying adding water, it's not like you take a bottle of water from the fridge and just pour it on some protein, it will start breaking up. Right? It's just that chemically, the hydrogen and the OH is returned to its original positions, and so the molecule is split. So, any questions, by the way? I feel like,
No, sir.
as usual, I'm just monologuing. So that is a peptide bond, right? Now this molecule is called a dipeptide. Di meaning you've got two of these guys, right? So if the number of units is greater than or equal to 3, then that would be called a polypeptide, right? But you don't generally you don't have a dipeptide going around or something as short as 3 to 10. Normally what you have are long chains of anywhere between 100 to thousands of amino acids long. And we're talking about one, right? One polypeptide chain could be this long generally, right? And then when we'll talk about that protein structure, and so you could write a protein molecule could be a single polypeptide chain. It could be more than one polypeptide chain coming together in a certain forming a certain structure and then doing its playing its part, carrying out its role. An example of a very small polypeptide is insulin. Right, so insulin is a very small polypeptide. It's only 51 amino acids. So it is considered to be a very small guy, right, as compared to all the other proteins that you have going around in the body. And insulin is a hormone. Right, something that we are familiar with. It's there in the regulation of the blood glucose level. Now, right, so far so good?
Yes, sir.
Okay. So next we're going to talk about bonds in proteins. And what do we mean by that? Oh, by the way, as I said, right, there's another term for proteins, and they can be called polypeptides. Right? Because of the peptide, obviously, named after the bond. So next up is the topic of the different bonds in protein molecules. And so what this is referring to, right, is not the peptide bond. The peptide bond is what makes the polypeptide chain. This is referring to all the other bonds, all other bonds or interactions between amino acid units. Right, and these could involve the amine and the carboxyl groups, right, and the R groups. So these are the three players. Right, now the way, as I mentioned, we're not talking about the peptide bond, so this bond is made, and this is not the one we are talking about, right? So, right, this is the peptide bond, and so we're talking about other interactions that these guys can have. So let's start counting them down. There are 3 of them mentioned. Let's go cross over here, that's not what we're talking about. So the first up is hydrogen bonds. Now we are familiar with hydrogen bonds from water, but water is not the only molecule that forms hydrogen bonds. So we know hydrogen bonding is something that occurs between an oxygen and a hydrogen atom, right, if they are polar, right? So if they are attached to other basically if they're coming in together somehow, or if they're attached to some other atom that is giving them polarity, they're giving a delta negative and delta positive charge, and so that makes them polar, and so that would give them the hydrogen bond. So in this case, if we look at these two guys, the hydrogen in the amine group, right? So nitrogen is right next to oxygen, and it does a similar thing, right? It's covalent bonds with the hydrogen, and it pulls the electrons in that covalent bond closer to the nitrogen, as because the only thing hydrogen has is a single proton, right? And so obviously its attraction is not strong at all compared to nitrogen, and so the nucleus, the protons in the nitrogen atom pull the electrons in the bond closer to the nitrogen atom, and that, because it has more of that electron charge, it develops a slight negative charge over here, and so I just mark one of them because the other one's utilized in the peptide bond, so it's no longer there, and so the hydrogen develops a positive charge. Similarly, right, the carboxyl ends, so you have right, and so this guy, right, if you have a free carboxyl end, you have a slight negative charge over here, right, and so the positive and the negative, right, attract each other with hydrogen bond. Obviously they're going to be closer by having drawn that, so just showing that the positive and negative charges, the polarities will attract, and right, that helps in maintaining the structure of the protein. Now individually hydrogen bonds are weak, and they are easily affected by pH and temperature, and that's why we know from our IGCSE that proteins are sensitive to pH, proteins are sensitive to changes in temperature, and one reason being is all the bonds that are affected, especially the hydrogen bonds, right? As I mentioned, we came across hydrogen bonds with water, and even there an individual hydrogen bond is weak. The reason why water has certain properties that are different from other similar molecules is because it has a lot of hydrogen bonding going on, right? And so you know, one is weak, but thousands and millions of weak bonds together, right, are strong. Just like the analogy, a single thread is easy to break, but a rope isn't, right? And ropes are made of threads, but put together they're stronger. So, right, that analogy fits. So hydrogen bonds are one of the bonds that occur. Right, they give the shape and they hold the protein together. Yeah. They can also occur between certain R groups. So it's not that it's only the amine and the carboxyl groups. Certain R groups can also form hydrogen bonds, and that makes a difference because when we talk about structure, whether it's the amine and carboxyl groups that are forming the hydrogen bonds or the R groups matters, right? Which one is it that's forming the hydrogen bond? Okay, moving on quickly, I want to cover this part in the current class. Disulfide bonds is the next one. And disulfide bonds basically are formed between two cysteine molecules. So cysteine is one, right? Is an amino acid. And so we are being particular over here that it has to be cysteine, and that's because it's the only amino acid whose R group has sulfur in it. And so what they have is, right, the R group is CH2, right, CH2SH. And so what happens, right, these guys are less, they form less frequently because they can only occur between the specific amino acid cysteine. So if there's two of them in close proximity in the amino acid chain, what happens is there's an oxidation reaction. There's the oxidation of each, and instead of having this, right, what you go on to have is a disulfide bond, right? So the hydrogens are removed, and we know, right, one definition of oxidation is the loss of hydrogen. So the hydrogens being removed from, right, the sulfur of each of the two cysteines, and right, there's a bond being formed between the two sulfurs directly. Now this is a covalent bond, so right, the disulfide bond is a covalent bond. So individually it's strong, right, much stronger than the hydrogen bonding, but the thing is that it is less frequent, right? So you only have a few of these appearing in a polypeptide chain. And again, right, they are important for folding and giving the shape to the protein. And finally, ionic bonds. Alright, so ionic bonds can form between some of the strong positive and negative amino acid side chains of the R groups. now, right, you've learned another term. The R groups can be called side chains. So if you ever come across that, you know what they're talking about. And right, they could be found deep inside the protein molecule. They are strong, right, ionic bonds, right. They're the strong bonds, but they're even less common. Right, and so an example they've given in the book is the the our hair. I don't know what's the proper word to use for it, but the texture or the style of our hair, whether we've got straight hair or wavy hair or curly hair or stuff like that. And so people who have curly or wavy hairs, they have disulfide bonds and certain ionic bonds going on. And all the chemicals or procedures using a curling iron or blow drying or certain chemicals that are used can change, they can affect the bond, sometimes temporarily and sometimes permanently, right? So giving yourself curls or straightening your hair by using heat is temporary. That changes the bonds, maybe breaks them, right? They could reform over time. And using chemicals can bring out permanent change. Now, permanent as in the hair that is present and is exposed. Obviously, hair grows, so the part that on which chemical was used, it's going to stay the way that it is. Whatever changes have been made as to its look or form. When it grows out, the new hair that will grow will be as it would be naturally, and so either that is treated to get the same look, or the older segment that's now at the farther end is cut off to remove that change. But just an example, right? So you can see, because otherwise you can't really see effects of these bonds, and so your hair is a very good example of that happening, or any other person you see with wavy or curly or just the ends curling, right, or something like that. Okay, I will stop here because the next section would be better dealt with in one continuous class. It talks about the structures, the structuring of a protein, of a polypeptide, right?
Yeah.
So any question?
No, sir.
All right then. I'll see you in next class then, and we'll wrap this up.
Sure.
Okay.
Yes.
All right.